Good afternoon. I am Dr. Georgette Lee. I will be sharing to you today the histology of the urinary system. So, the urinary system, the function of your kidneys is the regulation of balance between water and electrolytes. You have your inorganic ions, and the acid-base balance. It functions to excrete metabolic waste along with excess water and electrolytes in the urine. The kidney's excretory product, which passes through the ureters from temporary storage in the bladder before it's released to the exterior by the urethra. Your kidneys also serves to excrete many bioactive substances and many, including the drugs or medications that your patient takes. It also functions to secrete renin, a protease important for the regulation of blood pressure, by cleaving a circulating angiotensinogen to angiotensin 1. It also helps in the production of your red blood cells by the secretion of your erythropoietin. This is a glycoprotein growth factor that stimulates erythrocyte production in the red marrow when the blood O2 level is low. Your kidneys also help regulate your vitamin D and calcium because it is where the conversion of your steroid prohormone vitamin D that is initially produced in the skin to the active form to 1,25-dihydroxyvitamin D3 or your calcitriol, which in turn will help control your calcium by increasing absorption of calcium in the GI tract. Your kidneys also functions for gluconeogenesis during starvation or periods of prolonged fasting, making glucose from amino acids to supplement this process in the liver. So you have your kidney here, as you can see. It is 12 cm long, 6 cm wide, with 2.5 cm thick in adults. It has a hilum with a concave medial border where nerve enters and the ureter exits and blood and lymph vessels enter. It has a concave lateral surface, both covered by a thin fibrous capsule. So here is an illustration of your kidney where you can see the renal cortex, your renal medulla, and then each of your renal papilla, which is composed of your renal papilla, which will compose of your globe and a fibrous capsule. You have your minor and major calyx, your renal pelvis, your renal artery, and your renal vein. So your renal cortex is a darker stained region with many round corpuscles and tubules cross-section. Now your inner medulla or your renal medulla consists mostly of aligned linear tubules and ducts. It consists of 8 to 15 conical structures, called renal pyramids, separated from each other by the extensions of the cortex called your renal column. So your renal column, which is a portion of your cortex that separates each of your renal pyramids. The renal pyramids is mainly seen on your renal medulla. So a renal lobe is a renal pyramid plus a cortical tissue and its base and extending along its sides. So you have here your renal column on each of the sides and your renal pyramid. And the tip of each of the renal pyramid is called the renal papilla, which will project into a minor calyx that collects urine formed by the tubules in one renal lobe. So here you see an illustration of your nephron, which is your functional unit of the kidney. Each of the kidney contains at least 1 to 4 million of nephrons, and this consists of a renal corpuscle and a long simple renal epithelial renal tubule with 3 main parts along its length. So a renal corpuscle contains your glomerulus, your glomerular capsule, which has a visceral and a parietal layer, and a capsular space. Then you have your renal tubule, which will be divided into parts. So in a nephron, you have a renal corpuscle with a renal tubule. So the major different divisions of a nephron is your renal corpuscle or your glomerulus. So it's an initial dilated part enclosing a top of capillary loops, and the site of blood filtration always located in your cortex. Your renal corpuscle is always located in your cortex. A proximal tubule, this is a long convoluted part located entirely in the cortex with a shorter straight part that enters the medulla. You have your loop of Henry or your nephron loop, which is in the medulla with a thin descending and a thin ascending limb. A thin descending and a thick ascending limb. Distal tubule consists of a thick straight part ascending from the loop of Henle back into the cortex and a convoluted part completely in the cortex. And you have your connecting tubule, which is a short minor part linking the nephron to the collecting ducts. Your nephron is connecting tubules from several nephrons merged to form collecting tubules that emerge as larger collecting ducts. These converge into the renal papilla, where they deliver into the urine to a minor calyx. Cortical nephrons are located most completely in the cortex. So when you say cortical nephrons, both your renal corpuscle, so your glomerulus, your Bowman's space, your Bowman's capsule, with your renal tubules are all located in your renal cortex. Your juxtamedullary nephrons are the ones we have your 1/7 of the total of all of your nephrons that would lie close to the medulla and have long loops of Henle, because almost your cortex would just lie beneath the cortex and the medulla, with the projections of your renal tubule going down along the medulla. So the kidneys will receive 20% of your cardiac output in humans. It has less than 1% of your body mass. It filters 400 ml per 100 gram of tissue per minute. And it creates 1 to 1.2 liter, or filters 1 to 1.2 liters of minute per blood, and 1 ml, and produces 1 ml per minute of urine. So you have your left renal artery with your renal vein. So both, this is just an illustration of your both right and left kidneys with the aorta and your inferior vena cava. So this is the blood flow to the kidney. So from your renal artery, going to their segmental arteries, going to your interlobular arteries, to the arcuate artery, then to your afferent arterioles, which will then give way to your glomerular capillaries. Then after filtration, your afferent it will exit now the your glomerulus to your afferent arteriole, going to your peritubular capillaries or your vasa recta, then to your interlobular veins, to your arcuate veins, to the segmental veins, then to the renal vein. So here, again, just a different illustration. From renal artery, going to your segmental artery, going to the interlobular artery, then to your arcuate artery, interlobular artery, then going to the afferent arterial glomerulus, exiting out the kidney via the afferent arteriole, which will then be either going to your peritubular capillaries or vasa recta, which will now go to your interlobular vein, arcuate vein, interlobar vein, and then to the renal veins. So this is we'll just briefly discuss the renal function, filtration, and secretion and absorption. So your kidneys functions to filtrate filtration of water and soils in the blood. So blood will leave the vascular space and enter into the lumen of urine. Tubular secretion, substances from your epithelial cells of the tubule into the lumens, usually after uptake from the surrounding of your interstitium and capillaries. Your kidneys also have a capacity for tubular reabsorption. So substances from your tubular lumen will go directly or across the epithelium into the interstitium, back to the capillaries, back to your blood. And excretion. It happens along the length of the nephron tubule and collecting system. So this is your urine formation. So the filtrate receives various secreted molecules. When you say secreted, it's taken out of the blood, while others are reabsorbed and enters the Meyer calyces as your urine. So here is your renal corpuscle. Is composed mainly of your glomerulus with your Bowman's space and your Bowman's capsule. So blood will come in through the afferent arteriole, going inside your capsules or different capillary loops, and then going out to your efferent arteriole. So it is your renal corpuscle, your glomerulus is about 200 micrometer in diameter. It has a small mass of capillaries called the glomerulus with a bulbous glomerular capsule. So that's your Bowman's capsule, surrounded by a double-walled epithelial capsule called the glomerular capsule or the Bowman's capsule. Your Bowman's capsule has the podocytes. This is the internal or visceral layer of the capsule that closely envelopes your glomerular capillaries, and this is finely fenestrated. And also it has your parietal layer forms, which forms the surface of your capsule. So here in the picture on your left, you have in your right, you have here the PL, which is the parietal layer of your glomerular capsule. It's your Bowman's capsule, part of the Bowman's capsule, and then you also have a capsular space, and then in between or in the middle are your confluence of your capillary loops, which is your glomerulus. In each of your capillary loops will be lined by your podocytes, which is the internal or the visceral layer of the capsule that closely envelopes the glomerular capillaries. So we will have a clearer picture of this. So in between the two capsular layers, your parietal layer and the visceral layer, which is your podocytes, is a space which receives the fluid filtered to the capillary wall and the visceral layer. This is your capsular space or the urinary space, which is now your glomerulus have a vascular pole and a tubular pole. So literally, your vascular pole is the one that receives your afferent and the blood exit out to the efferent arteriole. Now, the tubular pole is where the secreted fluid comes out in your proximal convoluted tubule down to the other tubules. So, there's a flow of blood going to your afferent arterioles, going now inside your capillary loops, and then the filtrates or the ones that are filtrated from these capillary loops will go out, will go to your capsular space, exit your capsular space, and then going down to your proximal tubule, while the rest of the blood will go back or get out of your glomerulus through your efferent arteriole. So your renal corpuscle is lined, or the outer parietal layer of a glomerular capsule consists of a simple squamous epithelium supported externally by a basal lamina. And at the tubular pole, this will change to a simple cuboidal epithelium that continues and forms your proximal tubule. So here, this is not clearly seen. Only the afferent the vascular pole is clearly seen on the microscopic picture of your glomerulus. But in the visualization, in the picture, you will see that there's a transition between, look at your parietal layer of the glomerular capsule. This is lined by a simple squamous epithelium, and once it reaches the proximal or the tubular pole, this will in turn change into a simple cuboidal epithelium going to your proximal tubule. So here we will be a clearer picture of your podocytes. So this is a complete picture of your podocytes, an electron micrograph and an illustration of your podocytes. But you wouldn't be able to see podocytes this clear in under the microscopes. This is just an illustration. So again, the podocytes is a visceral layer of your renal corpuscle. It is unusual stellate epithelial cells together with the capillary endothelium or endothelial cells compose the apparatus for renal filtration. So here you see your podocytes with your pedicles, your filtration slits, and the podocyte body. So with the podocytes, it together with your capillary endothelial cells, it enables your glomerulus to perform its function for renal filtration. So from the cell body of each podocyte, it will elicit or have several primary processes that will extend and curve around the length of your glomerular capillary. Each processes will give rise to many parallel interdigitating secondary processes or your pedicles. And in between your pedicles are your filtration slit pores. These are elongated spaces in between the interdigitating pedicles around elongated space or filtration slits of 25 to 33 nanometer wide. You also have your slit diaphragms. Your slit diaphragms will span adjacent pedicles and bridging your slit pores. They are modified and specialized occluding or tight junctions composed of nephrines, other proteins, glycoproteins, and proteoglycans important for renal function. This surface is negatively charged. So it's important to know that it's negatively charged. Now here you have your glomerular basement membrane. So you have your visceral layer, which is your podocyte, and it will come to the glomerular basement membrane of each of your capillary loop. So this thickness is of 300 to 360 nanometer. This is the most substantial part of the filtration barrier that separates the blood from the capsular space, and it forms by fusion of the capillary and the podocyte produce basal laminae. So this is the illustration is an electron microscopy of your capillary loop. So you have here letter C, that's your capillaries. Now, on a zoom magnification, you will see a letter E here, that's your endothelial cell. Your BM stands for your basement membrane. So this is your glomerular basement membranes. And you will see you have the P here, that's your podocytes. That's the pedicles of your podocytes, which in between you have filtration slits. So you have your glomerular basement membrane, again, is composed of laminin and fibronectin. In this fused basement membrane bind integrins of both the podocyte and the endothelial cell membranes and the mesh cross-link type of type 4 collagen and large proteoglycan. Glomerular basement membranes restrict passage of your proteins larger than about 700 kilodaltons. So smaller proteins that are filtered from the plasma are degraded and the amino acids reabsorbed in the proximal tubules. So here is a better illustration. So this is the filtered blood and this is the blood that we have to filter and the filtered blood or the filtrate. So you have here glomerular filter, your fenestrated capillary endothelium, which is your basement membrane, under your basement membrane. Then you have your glomerular basement membrane, and then you have your filtration slit diaphragms in between pedicles, which will block large proteins. So you see, large proteins will not be able to get in or get out of your kidneys. Only small proteins are the ones that will be filtered in your blood. So this will be, so other things that will be filtered through the kidneys would include water, glucose, amino acid, ions, urea, hormones, vitamin B and C, ketones, and very small amounts of proteins. So the fenestration of the capillary endothelium, which blocks blood cells and platelets, the thick combined basal lamina, or the GBM, or your glomerular basement membrane, restricts large proteins and some organic ions. And the filtration slit diaphragms between pedicles, which will restrict some small proteins and organic ions. Again, now, in your glomerulus, or you're composed with different capillary tops. So here you see capillary tops, capillaries, capillaries, capillaries. In between these capillaries are your mesangial cells. So what is the significance of your mesangial cells? They resemble vascular pericytes in having contractile properties and producing components of an external lamella. They will often stain more darkly. They and their surrounding matrix comprise the mesangium, which fills interspaces between capillaries that lacks podocytes. Okay? So, you will see mesangial cells will be seen in between capillaries that lacks your podocytes. Okay? So, you will see here a capillary with the basement membrane, but the podocyte is not directly attached to the basement membrane. So, in between two capillaries, that's your mesangial cells. The functions of your mesangial cells, or the mesangium, it will physically support of your capillaries within the glomerulus. Adjusted contractions in response to blood pressure changes, which will help maintain the optimal filtration rate. It is or has a function of phagocytosis of proteins, aggregates adhering to the glomerular filter, including antibody complexes, abundant in many pathological conditions, secretion of several cytokines, prostaglandin, and other factors important for immune development. So here you have now your juxtaglomerular complex. Remember the vascular pole of your glomerulus. So you have your vascular and jugular fold. So the vascular pole is where your afferent and efferent arteriole comes in. So this is where your juxtaglomerular complex is. You have, in the juxtaglomerular complex, you have your macula densa. You have your juxtaglomerular cells. And you have your Lacey cells. So your macula densa, so here you will see clearly it is seen that you have your macula densa here. So you have again your glomerulus, the one in between, the one with the podocytes, your glomerulus. Your Bowman's capsule is the one that the outer layer. And then your urinary space, then your podocytes, your glomerular tuft. Now going, you know that this is your vascular pole because you have here your macula densa. And the macula densa lies on the distal connecting tubule. So that's DC. And then you have your juxtaglomerular cells. So this is a specialized region of your distal convoluted tubule. Cells are tall, columnar, closely packed nuclei, thin and discontinuous basal lamina, which will release renin. So this is the one that releases renin, macula densa. Remember that. You have your juxtaglomerular cells. So above your macula densa will be your juxtaglomerular cells, or adjacent to the macula densa. These are the epithelial cells in the tunica media. It's larger in size and internal elastic lamina is absent. Look at here. So the juxtaglomerular cells is congruent or lies in your near with your afferent arteriole. That's why it's important to remember that it is an epithelial cells in the tunica media of your afferent arteriole. Now you have your extraglomerular mesangial cells, which are your Lacey cells. They are structurally resemble intraglomerular cells, and function is not clear. So these are your Lacey cells. Now you have your proximal tubule. We're going now down to the tubular pole. You have your simple epithelial cuboidal epithelium. Simple cuboidal epithelium. The cells are well stained with numerous mitochondria, prominent basal folds, and lateral interdigitations. They have long microvilli. Lumens are often occluded and located at the cortex. Remember, your proximal convoluted tubule are always located at the cortex because your corpuscles, your renal corpuscles, are all at the cortex. Specialized for both reabsorption and secretion. It has a prominent brush border in the lumen that facilitates reabsorption. Many long basal membrane invaginations and larger interdigitations with neighboring cells. It reabsorbs of all, responsible for the reabsorption of all organic nutrients, all proteins, most water and electrolytes, and secretion of organic ions, cations like your hydrogen and your nitrogen or ammonia. Perform hydroxylation of vitamin D and release to the capillaries. Fibroblastic interstitial cells in the cortical areas near the proximal tubules produce erythropoietin. So in the proximal tubule happens the activation of your vitamin D to calcitriol, which will be then excreted to your which will be then released to your capillaries. Then you also the proximal tubule also performs the production of your erythropoietin. So this is a microscopic illustration of your tubules. So always remember that the one that nears or envelopes your glomerulus, or the ones that are near, would be your proximal tubule. So here this is clear. You have here your glomerulus, then you have your tubular pole, which will be, again, it's always filled. The proximal tubule doesn't have a clear lumen because it always looks dirty inside, and that's your proximal tubule. Now, adjacent or near your proximal tubules would be your distal tubules. So we will discuss this. So in order for you to distinguish between a proximal tubule and the distal tubule, because the distal tubule, remember, also sometimes will lie near your glomerulus at the vascular pole. But then, the main difference with them would be the inside of a proximal tubule will always look dirty, or it will always be filled. So you have now your loop of Henle, your thin limbs, the thin limb and a thin ascending limb. It has a simple squamous epithelia, few organelles located in the medulla, and the passive reabsorption of sodium and chloride. Now you have your loop of Henle, a thick ascending limb. This is simple cuboidal epithelium, no microvilli but many mitochondria. They have medulla and medullary layers, their active reabsorption of electrolytes. You have now your distal convoluted tubules. They are simple cuboidal epithelium cells, smaller than your proximal convoluted tubule. They have short microvilli and basolateral folds and more empty lumens. So they have fewer mitochondria. Located at the cortex also, performs reabsorption of electrolytes. The rate of sodium reabsorption here is regulated by your aldosterone from the adrenal glands. Now you will now have your collecting ducts, which will carry the filtrate into the collecting system to transport into the minor calyx. Your collecting ducts are formed by the extension of the connecting tubules, several joined together in the cortical medullary rays. They are simple cuboidal epithelium. The average diameter is 43 micrometer. At the medulla, these merge, forming larger and straighter collecting ducts, which increasingly, with columnar cells and overall diameter, reach to 200 micrometer, going to the papillary duct. So, in the collecting ducts, you have your principal cells, which are particularly rich. They are pale staining, few organelles, sparse microglia, and unusually distinct cell boundaries. The medullary collecting ducts are the final site of water reabsorption from your filtrates. It's located in the medulla and the medullary rays. Again, it regulates reabsorption of your water, regulates secretion of potassium, and they are particularly rich in aquaporin channels. Now, also in your collecting ducts, you have your intercalated cells. They are darker with more abundant mitochondria, responsible for the reabsorption of potassium, and also it helps and maintain acid-base balance. They will either secrete hydrogen ions or bicarbonate. So this is the cross-section of a medullary renal pyramid. Shows the simple squamous epithelium of your thin ascending limbs of Henle, and then its thick ascending limbs, which will be shown as letter A here, and then as well as your pale columnar cells of your collecting ducts. So here you will know you have your thin ascending limbs as lined by a simple squamous epithelium. Now your thick ascending limbs and your collecting ducts, which would be paler staining columnar cells. Again, just a different illustration. So this is an illustration. The orange one are your proximal convoluted tubules. And now the yellow ones would be your thin descending limb. Now also having a thin and a thick ascending limb. Then a distal tubule. And then your collecting ducts, the blue one. So now going down to the ureters, your bladder, and your urethra. So urine. So this is again the transport of your urine by the ureters from your renal pelvis to the urinary bladder, where it is stored until emptying by the micturition via the urethra. So your ureters have 3 layers. Again, this is your muscula, the mucosa, the muscularis, and your adventitia. So nearest the lumen would be your mucosa lining, which is lined by a uniquely stratified uroepithelium or transitional epithelium. The layers of your ureters are gradually becoming thicker closer to the bladder. So your mucosa layer with transitional epithelium. Now you have your lamina propria. After that, you go in to your muscular layer and then going to the adventitia. So the thick muscularis of the ureters move urine. Your muscular layer, look, is thick here because it is responsible for the peristaltic contractions of your ureter in order to move your urine from the kidneys going down to your bladder. It has prominent mucosal folds. So the muscular layer is the one, due to the peristaltic contractions, are the one that produces these mucosal folds when the lumen is empty. So the layers of your urothelium, they are single layers of small basal cells resting on a very thin basement membrane. These are an intermediate region containing from 1 to several layers of cuboidal or low columnar layer cells. There's a superficial layer of large bulbous or elliptical umbrella cells. So your bladder wall and your urothelium. So in here, in the pinkish picture, this is the neck of the bladder, which is near the urethra. So the wall shows 4 layers. You have again in the bladder your mucosa with your urothelium, that's a U. And then you have your LP, your lamina propria. And then you have a thin submucosa. Then you have an inner layer, a middle layer, and an outer layer of a smooth muscle cell. And then again your adventitia. So when the bladder is empty, so without urine, the mucosa is highly folded. And the urothelium has a bulbous umbrella cells. So you would see here, the letter U would have a bulbous umbrella cell. And then when the bladder is full, these folds will actually elongate. Look at the U here. At the pinkish portion, or a pinkly stained microscopic picture of your bladder, your urothelium, or the bladder is full, then the urothelium will be thinner. And your umbrella cells would be flattened. So the U is your umbrella cells. There are umbrella cells in the bladder. well-developed, extensive intercellular junctional complexes surrounding the unique apical membranes. Most of the apical surfaces consist of asymmetric unit membranes in which regions of the outer lipid layer appears containing mostly integral membrane proteins called uroplakins. Uroplakins acts as a osmotic barrier protecting its cells and the cells of the surrounding tissues from the hypertonic urine and preventing dilution of stored urine. Urothelium is surrounded by a folded lamina propria and submucosa, followed by a dense sheet of interwoven smooth muscular layers and endothecia. Your urinary bladder. The bladder's lamina propria and dense irregular connective tissue of the submucosa are highly vascularized. The bladder in an average adult can hold at least 400 to 600 ml of urine. And the urge to empty or to urinate, it will be start at 150 to 200 ml of urine. All the urinary passages are covered externally by an adventitial layer except the upper part of the bladder that is covered by a serous peritoneum. Your urinary bladder had a detrusor muscle, the musculus layer. There are three poorly delineated layers collectively called the detrusor muscle, which contract to empty the bladder. They're seen most distinctly at the neck of the bladder near the urethra. All the urinary passages are covered externally by an adventitial layer except for the upper part of the bladder that is covered by a serous peritoneum. So the urethra is a tube that carries the urine from the bladder to the exterior. Urethral mucosa has prominent longitudinal folds, giving it a distinct appearance in the cross-section. In men, the two ducts of sperm transports during the ejaculation join the urethra at the prostate gland. The middle part of the urethra in both sexes is surrounded by external striated muscular sphincter. So a male urethra, this is longer and consists of three segments. You have your prostatic urethra, which will extend the prostate gland, lined by the urothelium. A membranous urethra is a short segment, passes through the external sphincter of the striated muscle and is lined by stratified columnar and pseudostratified columnar endothelium. And your spongy urethra, which is about 15 cm in length, is closed within erectile tissue of the penis, lined by your stratified columnar and pseudostratified columnar epithelium. Female urethra is a urinary organ, 3 to 5 cm long, initially with transitional epithelium, which transitions to non-keratinized stratified squamous epithelium, continues with that of the skin at the labia minora. So here is an example of your urethra. It is a fibromuscular tube that carries urine from the bladder to the exterior of the body. So letter L would be your lumen. So you have your a mucosa that has large longitudinal folds around your lumen. At higher magnification, you will see that there are areas of unusually stratified columnar nature of the urothelium. It has thick epithelial lining, varies between stratified columnar in some areas and pseudostratified columnar at the distal end of the urethra. That is it for my lecture today. Thank you.